Alright everyone, welcome to the stream. We're gonna begin in a couple minutes. And then everyone's silent, okay.
Well, hello.
Yeah, we're letting you do your thing.
Oh, okay. Well, I've never really done this before, so it's new.
Okay, I'm back. I was looking at the lasagna.
Miguel, why is your profile picture so odd?
That's
It's
a
a
dog,
dog.
isn't it? Alright everyone, welcome to the stream.
Oh,
We're gonna begin in a couple minutes.
still odd.
Jerk.
I'm sorry. Too bad we forgot to get music for this. Alex.
Music is Well, I'm sorry. I would have gotten music if my computer didn't, like, die.
Oh, okay.
Tots. Okay. God, I'm tired.
Music for this.
Trevor's like, the voice of God has spoken. Music is Well, I'm sorry. I would have gotten music if my computer
Hey, I
didn't,
turned off the
like, die.
volume of the stream, but it is so delayed.
It is. It's about 45 seconds, I think. But there's nothing we can do about it.
Yeah, there's nothing.
I'm just gonna
Look,
turn
it's
the
Miss
volume
Anderson.
off on, like,
Hi, Miss Anderson.
What?
Hi, Miss Anderson.
What are you talking about? Am I not
The
seeing
chat.
the chat break?
My chat is delayed. I don't know about you guys. Music for this.
No,
Trevor's
it's
like the voice of
it's
God
mainly
has
the
spoken. Music.
in my opinion.
The last message I see is Trevor saying the voice of God has spoken.
Yeah, that's the last one I saw too.
Has something else been said?
Yes. It's like Miss Anderson's like, hi guys.
Oh.
Maybe I should, I might appear at some point.
I'm just gonna turn off the. Yeah, I'm gonna keep the volume on YouTube turned off just so that I don't get confused between the delayed voices.
Hopefully it actually is Miss Henderson and it's not someone who made an account called Henderson.
Oh, there are quite. There are a lot of trolls out there.
Because I could see someone doing that. Why does this computer have to be so terrible?
Shane's like working on his computer.
Wait, so 3:10, you're starting your thing, Ravi?
Yes.
Yeah, we're gonna start at 3:10.
Okay.
So wait, is it turned off? Can they not hear us or?
They can hear us.
Oh.
Of course it's delayed, 45 seconds, so they'll hear you in 45 seconds.
That's quite a bit of delay.
Yeah, YouTube transcoding servers, we can't do anything about it.
Which isn't much better, it's like 30 seconds.
So basically, they'll just see the stream like 45 seconds after we say it.
Yeah.
Yes, but to them it doesn't matter because
I know,
they're...
because they can't. They're not from our perspective. They don't
Yeah.
see the delay.
Yeah.
Hello, 45 seconds into the future.
I really hope nothing bad happens in this chat, because the chat's being stupid. So... What the...
Hopefully my computer doesn't blue screen again. That would be terrible.
My computer's probably gonna blue screen. A text message.
Oh, yeah. Probably turn this on.
Yeah, see, Mia asked, how do we know you are the real Miss Henderson? Like, do we have confirmation that's Miss Henderson?
I don't think anyone would want to spend the time to make a Miss Henderson account. And
Trust me, people would.
Isn't Miss Henderson married?
Oh my god! It's already suspicious.
Alright, it's 3:10. So you guys want to get started?
Let's do this thing.
Alright.
Oh.
Okay, so welcome to the stream, everyone. Hopefully you have pencil and paper so you can write down some notes or things that you might want to go back and review. Of course, we'll be releasing this PowerPoint afterwards so you guys can look at it. Right,
also, the stream will be a video afterwards.
Yes, you can rewatch it afterwards. Alright, so let's begin. Oh no. Alright, so today it's going to be molecular and cellular biology, and then we're going to do plants as well, and then tomorrow it'll be human body, and then evolution.
With me.
Yeah. So,
I still
okay.
find a hilarious Shane in the background.
Oh yeah. So, what are cells? Well, the cell is the smallest unit of life that can carry out all the functions of a living thing. They can also vary in size, shape, and structure, and a cell's shape is related to its function. That's pretty straightforward. Okay, so what is cell theory? Cell theory was created by a couple of scientists. I don't know the names of the scientists. Any of you guys remember?
Nope.
Nope. Okay. Well,
That helps, right?
yeah, I don't think you need to know the names of scientists, but basically what they came up with is all living things are made up of one or more cells, so you can't be a human and only be consisting of one cell. That would be strange. Cells are the basic units of structure and function in organisms, and then all cells come from existing cells. So you guys can think about...
Isn't that the cell theory?
That is cell. Yes, Alex. All cells come from existing cells. So if you want to create a new cell, it would have to come from another cell. They can't come from any other place. And then there's two types of cells. There's prokaryotic and eukaryotic. And then prokaryotic is bacteria, and then eukaryotic is any other cell, like animal cells and plant cells. And an example would be like a red blood cell in humans. Prokaryote, there's no nucleus. Example is bacteria. There are no organelles, so you don't have any like mitochondria or ribosomes or anything. It's small. 15. You guys know what is?
That is...
It
I
doesn't.
think it's like an atomic unit. It's a
nanometers.
UN. That's fine.
Oh,
Yeah.
and also,
I don't
if
think it's
you
a
guys,
U. I think it's like the nano symbol.
Alright. And then they have a
Ravi.
simple structure. Yes, Alex?
I just want to tell the people, if they have questions along the way, just type it and we'll eventually get to it.
Yeah. Alright. And then simple structure, which is basically it's a ring of DNA in the middle and then the membrane on the outside. Eukaryote, it has a nucleus. Plant and animals are made up of eukaryotic cells, and then they have organelles, which is mitochondria and ribosomes. They're 1,000 times larger than prokaryotes, prokaryotic cells, and they have a complex structure.
An easy way to remember this is that with it being made of plants and animals, for the difference between euc and prokaryotic cells, is we think of Zach Euchre having eukaryotic cells. That's an easy way to remember.
Remember Zach. Just remember Zach.
Alright, so plants versus animal cells. So plants have certain organelles that animals lack. Organelles are kind of like the parts of the cell. So the plant has a cell wall, a rigid outer covering that protects the cell and helps it hold its shape. And it has chloroplast full of chlorophyll, and that's the photosynthetic pigment. A vacuole, it's a large central compartment. It absorbs water, ions, or waste products. So this is a visual representation. So as you can see, there's a central vacuole in the plant cell. That's I believe that's an orange on my screen. No, that's the white on my screen. Sorry. Those are the vacuoles, and then the cell wall is on the outside, and you can see that the animal cell doesn't have it. Let's see. Is that the only differences, guys?
For what?
For plant versus animal cells? Yeah, cell wall, vacuole, and chloroplast, which are the photosynthetic pigment.
For some reason, I always remember mostly just the cell wall part,
And
which I know isn't
vacuole.
helpful, but like I don't know. Like if you see cell wall in any cell, it's pretty much going to be a plant cell and not an animal cell. Just like an easy way to distinguish it, because that's usually how it's shown.
Yeah, cell wall, and also I remember vacuoles and cell wall. Usually I don't remember chloroplast, but that's kind of a given if it's a plant. Alright, so we have cell division. Cell division there's two types. There's mitosis and meiosis. Mitosis is pretty much any body cell that replicates itself, and meiosis is cells that create sex cells. It's like sperm and the egg. Cell size factors. So this is why cells divide. The surface area of the cell membrane for molecule movement. As cells increase in size, their volume increases faster than their surface area, so they need to split into more cells. Controlling influence of nucleus only extends so far. So the nucleus, if the cell gets too big, it's not going to be able to control everything. So you're going to have to, the cell is going to have to split. Therefore, the cells need to stay small. So there's 4 parts, and then there's one more afterwards. But how we remember the 4 parts is PMAT, prophase, metaphase, anaphase, and telophase. And then there's cytokinesis afterwards.
Ravi?
Yes?
Cameron said, don't animal cells have vacuoles as well? They are just smaller?
The plant cells has a large central vacuole, and the
Do animal cells have any equivalent?
Can't remember. I pulled this off of the PowerPoint.
Yes, they have smaller ones.
Oh yeah,
Oh, they
sorry,
do?
correction guys. We don't know everything, so there might be a couple mistakes. Okay, so why do cells divide? It's to increase in size, so increase the number of cells, so they can differentiate and have a specialization. Repair, level of repair or regeneration varies, and then in humans, it's low, and C stars, it's high. A C star is just an example, because if you cut a C star, it'll eventually grow into another C star from, say, if you cut off one of its limbs or arms. So this is the parts of mitosis. In interphase, materials necessary for cell growth and division are produced, and this takes up 90% of mitosis.
This is, sorry if I intrude, but the interphase is basically the part where cells grow and gain nutrition and stuff like that, and it's right before we hit mitosis.
Oh yeah, sorry. So there's 3 phases: gap 1, synthesis, and gap 2. Gap 1 is growth and development. Synthesis is chromosomes in the nucleus replicate, and then gap 2 is the cell manufactures organelles and the centrioles replicate, and then chromosomes stay in the loose form of chromatin throughout interphase. So chromatin is comprised of protein, RNA, and DNA. And then this is how we remember the stages, PMAT, and then cytokinesis. So it's prophase, metaphase, anaphase, and telophase. And then cytokinesis, we'll get into that in the next slide. So the first part is prophase. Chromatin, which I said is comprised of protein, RNA, and DNA. They condense into chromosomes comprised of two sister chromatids joined by a centromere. So the centromere is what holds the two sister chromatids together. We don't have a picture right now, but if you've seen a chromosome, you can see that it's at the middle, it's attached by a centromere. And then in prophase, mitotic spindle forms, the nuclear envelope dissolves, and spindle fibers attach to the centromeres. In metaphase, chromosomes line up in the middle of the cell, and they create the metaphase plate. In anaphase, centromeres divide. Spindle fibers pull one set of chromatids to each pole of the cell, dividing genetic material in half. Telophase, daughter nuclei are formed. A new nuclear envelope is formed. Chromosomes loosen back into chromatin. The mitotic spindle dissolves, and two nucleoli are formed. Cytokinesis. Cytokinesis is the division into two cells after telophase. Division of the cytoplasm begins during telophase. Sorry, I said after. Plants, the cell plate forms in the center of the cell during anaphase, which forms a new cell wall. In animals, the cell membrane folds inward to form a cleavage furrow, which eventually pinches the cell in half. And then you can see here in plants and animal cells. Prokaryotes, since prokaryotes don't have a nucleus, it can't perform mitosis, so it must do so by binary fission. So what happens is the central circle of DNA replicates, and then cytokinesis happens, and that's how it replicates. It has a similar cell cycle of growth, DNA replication, preparation, cytoplasm division, and it's faster because of its simpler structure. And this is how food spoils so quickly, and septicemia kills quickly.
I'm presuming we don't need to know what septicemia is.
No.
Okay.
Meiosis. Meiosis is a special form of cell division prior to sexual reproduction in which the number of chromosomes is reduced. There is two types of cells. There's diploid and haploid. Diploid cells have two sets of chromosomes, which is 46 or 23 pairs, and then haploid has 23.
Haploid's at 23.
Yeah, 23.
It's the basic way to put it is haploid cells are usually sex cells, and diploid cells are everything else.
Yes. And these are the stages of meiosis. It's just a picture. We'll come back to this. So for sex cells, they become haploid gametes. This is so the chromosome number can be reduced, because you get one haploid for your mom and one haploid from your dad, and that will give you a diploid cell. And this preserves the chromosome number of the organism instead of doubling it with each generation, which it'd be, how to explain this? It wouldn't be good if you doubled your chromosome number every generation.
Ravi?
Yeah.
My question is, what does the nucleus have to do with mitosis again?
What does the nucleus have to do with mitosis?
It doesn't have any special functions.
Oh, well,
in
during the
mitosis,
synthesis
does it?
phase, we know that the nucleus houses your chromosomes. So
Oh.
before your cells have to split, your DNA has to replicate.
Yeah,
Okay.
and since prokaryotes don't have a nucleus, it's not able to perform synthesis. Okay, so meiosis. During meiosis, chromosomes in each homologous pair replicate, and a single copy from each pair is going to each daughter nucleus. In humans, one diploid cell of 46 chromosomes, which is 23 pairs, yields 4 haploid cells of 23 chromosomes, 23 singles. It's totally random, in which each haploid cell ends up with a different variation of chromosomes. Independent assortment is random, which chromosome mom or dad's ends up in a gamete. So each of the 4 haploid cells will have a different combination of chromosomes. Eventually, the organism's diploid reproductive cells, 2N, undergo meiosis to form the next generation of haploid cells. So diploid cells create 4 haploid gametes. The timing of meiosis and mitosis can vary among different types of organisms, mosses versus mammals. The matching pairs of chromosomes in a diploid cell are called homologous pairs, one from mom, one from dad. These are identical except for possibly the XY pairing of the chromosomes in males. Jacob, could you explain this better, or?
Let me read over again. Oh, okay. What it means by this is that, the reason why they're homologous is because it has one of each side of the chromosome, I'm pretty sure.
Mm-hmm.
And, they're all identical, but the reason why it's different with males and females is because with a male, it's an XY pairing, and with a female, I believe it is an XX pairing, and with the size difference, the Y is a lot smaller than the X.
Okay. And then there's stages of meiosis. So since meiosis creates 4 cells instead of one new one, like mitosis, it must go through 2 stages. There's meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis. And then it does another set, which is meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2, and then cytokinesis again. So you end up with 4 cells, each with 23 chromosomes. So the first time, it's almost the same as mitosis, where it creates another cell with 46 chromosomes in it, but then it splits again through the second set, and that gives you 23 chromosomes. It doesn't replicate again, I believe,
Robbie?
right? Yeah,
Can Shane turn his camera off?
I think you got it.
Okay. He just doesn't. He's not really. Okay, there we go.
Okay, meiosis versus mitosis. They're both forms of cell division, and they involve similar steps: replication, dissolution of nucleus, and chromosome movement. And how are they different? In meiosis, daughter cells have half the chromosome number as parent cells. Daughter cells are produced by meiosis and are not identical to each other. That's random assortment of chromosomes in the tetrad. Jacob, do you remember what the tetrad was?
Honestly, no, I do not.
Oh, yeah, okay, me neither. You can always write down notes or questions for Miss Henderson, because we're not certified teachers. And
We try.
yeah, we try. In meiosis, four daughter cells are created versus 2 in mitosis. So, why sexual reproduction? Sexual reproduction is different from asexual reproduction because asexual reproduction doesn't allow the organism to adapt to its environment. Because asexual, I believe it's an identical organism is created, whereas in sexual reproduction, the chromosomes are varied, so you end up with a different combination of chromosomes. Here it says, random assortment of chromosomes during meiosis plus the random variation of sperm with egg creates variation in a population. In a changing environment, this diversity gives a species survival advantages. Okay, so cancer. When a single cell begins to reproduce aggressively and it invades other tissues, that's considered cancer. It is not a foreign invader, so it's not coming from the outside, it's your own cells, and it's slow to trigger the body's immune system. Do you have anything to add, Jacob or Miguel?
Well, when a cell, like if you're in biology, you know that when these cells continuously divide, they'll be, I guess they can, eventually they become cancerous, and they can start to spread to other parts of your body. So, let's say that you had a cancer in an organ, and it spread to your bloodstream somehow. It could go all throughout your body, and so eventually it would take over all the tissues, and that's how cancer basically kills you.
Okay.
That's all I can add.
Miss Henderson says you're doing great.
Oh,
Yeah.
thank you, Miss Anderson.
Thank you.
She'll hear this 45 seconds later.
Later, yeah.
This is why it's a little bit confusing.
Oh yeah,
Yeah.
the tetrad.
I'm not
thing
seeing the
in the
Oh.
chat.
PowerPoint like
What's
at the same
tetrad,
rate.
Miguel?
I have it.
Oh,
on
he
the
put it in the
chat, so-
he put in the chat. A group
Oh.
of 4 cells produced by successive divisions of a mother cell. Can you
Okay.
make sense of that?
No.
Oh, it's Oh, I think I was looking it up. A tetrad is basically where like it's 4 homologous chromatids or 2 homologous chromosomes, and basically what they do is this is when they start to touch and cross over, so that the so that the individual parts, so that the chromosomes mix and we get that variation in each gamete. It's basically where they touch over and they switch up.
Okay,
That's the best I can explain it.
that's good enough. Now we're moving on to transport. So there's 2 types of transport, passive and active transport. This is how stuff gets in and out of the cell. For passive transport, the movement of a substance across a cell without an input of energy. Active transport is with the input of energy. Wrong one. Alright, so passive transport, an example is diffusion, random movement of molecules from an area of higher concentration to an area of lower concentration. So it's down a concentration gradient until they reach equilibrium. This rate depends on temperature, size of molecules, and magnitude of the gradient. So this is an example of the diffusion of sugar, and you can see how it's a lump of sugar and then it divides into the solution.
Okay, that's not a good thing.
Facilitated diffusion. What happened? Miguel.
Stream is going in and out apparently.
At least that's what Ndoni says.
Yeah.
No one else has complained.
Let me check. Open Broadcaster says it's fine.
Let's ask the chat.
Just keep going until until like a lot of people tell us there's something wrong. Just continue.
Okay. There's facilitated diffusion. Carrier proteins help molecules across cell membranes. It's the same process of diffusion, but at a faster rate, and with large molecules or ions like glucose or and is sodium. And then there's osmosis, the diffusion of water across a semi-permeable membrane via aquaporin proteins. I believe roots use osmosis, right?
From like high concentration to low, right? That's pretty much it.
Yeah. So water moves down a concentrating gradient, high water to low water, or a low solute to a high solute. The different concentrations inside and outside a cell can cause water to enter or leave a cell, salted fish, or resurrection fern. Oh no, Miguel, we didn't crop this picture, right? But okay. So,
Not. Oops.
yeah.
Looks fine.
Okay. Oh, we'll just skip over that. There's 3 types of solutions. Hypertonic, in which the cell shrinks because the inside water percentage is greater than the outside water percentage. Isotonic, it stays the same. So the inside percentage is the same as the outside percentage. And hypotonic, in which the cell swells or kind of gets bigger. The inside water percentage is lower than the outside. And then we have active transport. So it requires energy, it requires an input of energy. It often involves carrier proteins. So the carrier proteins use energy to pump ions and molecules across the membrane, and transport of molecules against a concentration gradient low to high. So that's why energy is required.
Yeah, they're saying the stream's fine, so it must just be Andoni having issues.
Oh, okay. Examples of active transport. So the sodium-potassium pump in animal cells. Sodium is kept high outside the cell, and potassium is kept high inside the cell to allow the import of sugar, the contraction of muscles, transmission of nerve impulses, and the absorption of nutrients in plant roots. Okay, exocytosis. There's endo and exocytosis. We're going to do endocytosis in a second. So wastes and cell products packaged by Golgi into vesicles. These vesicles are kind of like the circles that you see on the screen. They then fuse into the membrane, and the materials are secreted out of the cell. This is how the cell transports things in and out. I guess waste. Is there any examples of things that are transported in and out via exocytosis? Is it only waste?
I think there's also nutrients and stuff.
Oh, okay.
In and out. Well, exocytosis.
Let me give a way to remember between the two. Endo means inside, so like so endo equals inside, and then exo equals outside.
Yes.
So remember that. So like to expel something.
Mm-hmm.
And then
Ravi.
Yeah.
Someone says, like, is PMAT a good way to remember prophase, metaphase, anaphase,
yes,
and telophase?
that's the way
Yes.
I remember it.
That's in the correct order?
PMAT,
Yeah,
yes.
PMAT.
And then
Okay.
cytokinesis.
Yeah. But I don't
That's-
think that's one of the, like, main stages it usually asks for.
Well, there's interphase, and then the actual mitosis would be
Yeah.
PMAT.
Yeah, so, like,
That's mitosis
interphase
itself.
and cytokinesis, they're kind of like, they're most...
That's cell division in general.
Yeah, but they're not technically part of mitosis.
No. No.
Okay.
Okay. So, endocytosis. So the cell membrane surrounds a molecule outside of the cell. A vesicle is formed around the molecule, and the vesicle moves. into the cell. And we have receptor proteins. These proteins are embedded in the cell membrane, and they detect and bind to signal molecules, which are carrying information. Once the signal molecule binds to the receptor protein, there's a change in the permeability of the cell membrane, and it forms a second messenger inside the cell, and the receptor acts as an enzyme. Do you have an easy way to say this, Jacob?
Not really.
Okay.
I do not.
All questions go to Miss Henderson. Alright, structures for movement. These are extensions of the cytoskeleton. There's two types. There's cilia and flagella. Cilia are short hair-like, more than 5 per cell, and they have an oar-like movement. Flagella are long, tail-like, 1 or 2 per cell, propeller-like movement in prokaryotes, and whip-like movement in eukaryotes.
I think cilia is for the egg cell and flagella is for the sperm.
Yeah.
So
The word flagella is so entertaining.
in
The
my
way-
class, every time they say flagella, everyone like ninth grader named Tyler just keeps repeating.
Oh,
it over.
Tyler.
I think the easy way. This is how I remembered it because Miss Henderson did this. But think of cilia being very soft, very calm, flowing through stuff, and then flagella just with like.
We're quoting Miss Henderson.
So yeah, I guess
Words
that's an
from
easy
Miss
way to
Henderson.
remember it. Since flagella is like kind of a bit more
Flagella.
like harder to say, I guess. Think of it as more like rapid, while cilia is sort of like easy to say, so it's like calm. I guess.
Flagella can move a little bit faster, but cilia can move in direction. Like
Shane
directions.
says
More directions.
flagella moves like a flag in the wind, because the first
Flag.
4 letters are flag.
That works.
Actually,
Oh.
that's actually, yeah, that does work, I guess.
Alright, next slide. Alright, DNA replication.
I'm done. I'll give it to you.
Alright, thank you.
Alright, just one moment, please.
You can continue, Ravi, even though they're sending the file.
Yeah,
What's
but
in the file,
Jacob's
Robbie?
gonna explain this.
Oh, okay.
Okay. Oh god,
Where are
the
we?
pressure's on.
I'm not really a
Just-
big help in DNA stuff, so.
Yeah.
Oh, deoxyribonucleic acid? I got this.
Yes.
Okay. Do you want me to explain this, Robbie?
Yeah,
I guess
I'm on
we'll
the-
take a
I'm on
unofficial
slide.
break as it downloads.
Oh, okay.
Put a break on it, Ravi.
Alright, everyone, we're gonna take a break real quick. I know. We'll be back in 5 minutes. We're back.
Heyo.
Hello.
Hi, people.
Jacob sounded depressing.
You can hear us, right?
Yeah.
Alright. So, DNA replication. So, should I start explaining? Yeah?
Yeah. Go.
Okay. DNA replication, between gap 1 and gap 2, is the synthesis phase. And this is basically where your DNA starts to replicate. Now, DNA is known as deoxyribonucleic acid, and it is the molecule of heredity, so it stores all of the chemical information that makes you, you. It's the master plan, it is your blueprint. It is used to construct your proteins, and that's with protein synthesis. Next slide.
Yep.
Okay, so DNA replication. Like we said, it occurs during the synthesis phase of interphase, and it's carried out in multiple steps with different enzymes like helicase and DNA polymerase, but we'll get to that in a little bit.
Yeah.
Now, the nucleotides, the two halves of the DNA, they're anti-parallel, which means one runs one way, and then the other one runs the other way. They're called anti-parallel. At the end of the duplication, there will be two molecules of DNA, and we'll have it'll be made of one old strand and one new strand. So the actual process. Next slide. Good. So it starts off with a enzyme known as DNA helicase, and helicase splits your DNA into the two segments, into the two replication forks. By the way, something to remember: if it ends in ACE, that means it's an enzyme. So ASE, remember that. So it's with prokaryotes. It's a ring of DNA, and it will start at one point in the prokaryote, and it'll swing around until it completely splits it into two. With eukaryotes, it'll start at many points. So what happens is the helicase splits the DNA into two, and then we have RNA primase, and it'll add a small section of RNA into the open areas of DNA. Now, what the polymerase does is it joins the matching free nucleotides to the DNA templates. And the leading strand is 5 to 3, and it is made continuously, so without stops. But with the lagging strand, it has to do all of it backwards. Then it goes from 3 to 5. Now, protein synthesis. Did everybody get that?
Wait, can I say something?
Yes, you may.
I'm looking here. It says, isn't the Okazaki fragment that leftover part or something?
No. The Okazaki fragments is small It's like fragments of RNA, and they come in backwards and kind of awkwardly as compared to the leading strand, which is just a continuous zip of it.
I'm trying to remember.
Oh,
Is it anti-parallel or something? It goes up and down.
Yes. That's why it has a leading and a lagging strand.
Mm-hmm.
One runs from 3 to 5, and the other one goes from 5 to 3.
Leading is 5 to 3 and lagging is 3 to 5. Okay.
Yep. Protein synthesis. So how we get from those
How
from those
DNA
bases
creates
into making.
proteins.
Yes, it
Right.
does. But basically, we're taking the plans of your DNA and turning it into a person or a potato or a goat. So, it has your two main parts. We have the transcription and the translation. That would be the difference between writing your notes and then writing your notes in Spanish. We go from DNA to mRNA.
Messenger
And
RNA.
that's called your messenger ribonucleic acid, and this is what is going to start off going into the nucleus. So let's go to the transcription slide.
Yeah. How I remember transcription and translation, because sometimes you can get them mixed up. Translation has late in it, and that's how I remember it's the second part.
So we start off from our master plan, which is our DNA information, and we go to mRNA. Now this is your messenger RNA, and the messenger RNA is that long line of codons, and how the process works is we have RNA polymerase separate or unzip a section of the DNA. So let's say that we're coding for hemoglobin for blood cells. It'll work kind of like helicase in the fact that it'll unzip a section of your DNA so that mRNA can come in. Now, what it does is the RNA polymerase binds to a free nucleotide to their complementary bases on the DNA strand, forming your strand of mRNA. So let's say that we had, I'm trying to think, because I know that AUG is methionine. Well, basically, it'll do what the other side of the DNA would have, and I think instead of a T, instead of having thymine, it'll have uracil, right?
Yeah.
Yeah. But what it'll do is it'll completely, the mRNA comes in, it copies the information, and then until it reaches a section of DNA that's basically a stop, and then it's made of introns and exons. This isn't explained on here, but it has introns and exons, and I believe it keeps your exons and releases your introns.
Yes.
So introns are basically the stuff it doesn't need to make that protein, but the exons are everything that you do need. So next slide. This is a visual representation. If you look.
Oh wait, a good way also, like in my opinion, to remember translation is to because translation, it's kind of turning the amino acids into proteins as sort of like translating them. Like, I don't know, if you just think of the definition of translation, it's pretty simple to remember. If you see that the messenger RNA is sort of translating the amino acids or reading them into something, think of it that way. That's how I remember it.
We're in step 4, right? Showing step 4.
Sorry?
I accidentally skipped that, but it says the amino acids are brought in at one at a time, and it's known as, that's your tRNA, the little keys that have each amino acid, and there's always 2 tRNAs on a ribosome at a time. So your mRNA will whoa, wait a minute. I think I am.
I think it's supposed to be 3.
Guys.
Wait, always 2 tRNAs. Never mind.
Yeah, I've been skipping over some stuff. Sorry, guys.
It's okay. Where do I need to be for this slide?
Where it says translation.
Oh, okay.
I was going ahead a little bit. I was wondering, it's missing some stuff here. So, translation. Translation is the part where we start to make the protein. So, we take our amino acids and we turn them into proteins. So, mRNA is attached to a ribosome, and it's stuck between a ribosome and rRNA. Now, ribosomes can be found in the rough endoplasmic reticulum, or it can be found floating throughout the cell. A group of 3 nucleotides, so like your A, your U's, and your G's, is called a codon, 3 of them, and it's read at 3 at a time. And there are 64 possible codons. And each tRNA has a specific key so that it can read each codon, and with its matching anticodon. And so it comes to the ribosome, it matches up with its codon, and it brings its amino acid with it. Next slide, we're 4, 5, and 6. Each amino acid is brought one at a time on each tRNA, and eventually it'll make a chain of amino acids called a polypeptide, I'm pretty sure.
Polypeptide chain.
Okay, it's ATGC, but transcription makes it AUGC. ATG. Actually, it would be A to... Oh, it's A to TGC, but transcription is... Yes. Yes, it is. So... Yes. There will always be 2 tRNAs on a ribosome at a time, and this is so that the amino acids can continuously join together. With codons, it'll always, most of the time, 9 times out of 10, it'll start at AUG, which is methionine or methionine, and then it'll stop at a stop codon, and that'll be like UAA, UAG, or UGA. And then we have a visual representation of this, where it shows it being, the DNA being split, copied by the mRNA, going to the ribosome, being transcribed, or being translated, and then it's making the amino acid chain where it swings off and makes the protein. So, next slide, so slide 50 if you will.
Mutations.
So, mutations. So,
you want me to do this one?
We can both explain it.
Alright. So there's 2 types of mutations? Is there one?
Yes, frame shifts and point mutation.
So, mutation can happen in which, in the codon, there's, there's 3 letters, and then frame shift is when a letter is deleted, I believe.
It's either deleted or inserted, and it's a little more devastating, because instead of saying we have, we'll say, I'm trying to think of a way to put this, like, let's say that we had ATGCCGAT, I'll put that in there. If one is deleted, instead of being read ATG, CCG, and then AT, and it would probably have a last one because it's read 3 at a time. If one was deleted, it would be shifted all the way over. That's why it's called frame shift, and it would read it in a different frame, completely messing up the protein that it's making.
Yeah,
Robbie,
so every...
check SketchUp.
Oh, okay. Every one after that is changed, I believe. After the shift.
And it's read completely differently. And then point mutation is where it'll only change at a specific nucleotide base pair. So only one codon is affected. Now, it can still produce the same amino acid, but it might not always. If it's closer to the front of the 3 nucleotides, it can do a more devastating effect when it's reading it.
Ravi, did you get my message?
Yeah, got it.
Okay.
Right, you can do the internal controls, which you'll probably be better at.
Okay, internal controls. So these are signaling molecules made of cyclin proteins and CDK enzymes. Cyclin-dependent kinases. And then they control the timing and rate of cell division, skin versus nerve, and broken bones. At the end of each stage of the cell cycle, a checkpoint controls whether or not the cell will continue on to the next stage. So that would be from, say, prophase to metaphase, right?
Yep.
And there's a checkpoint, and that's when the signaling molecules will decide if the cell can go on to the next stage or no, and then it has to perform apoptosis, which is cell death.
Cell suicide.
Yeah, cell suicide. And external controls in humans, growth hormones produced in the pituitary gland near the brain, or external controllers of external controllers of rate and timing of cell cycle. So during puberty, lots of hormones, lots of cell division and growth. So a malfunctioning pituitary gland would could cause dwarfism and can be relieved by injections of human growth hormones, synthetic source invented by genetic engineering to eliminate disease transmission from human source. So what growth hormones do is they basically, during puberty, they cause you to grow. I'm pretty sure that's an easy way to explain it, right?
Yeah.
So
Dumb it down a
that
bit.
means that Zach has a lot of growth hormones, me, not so much.
Same here.
Okay, now on to carbohydrates and proteins. Jacob, you want to explain this one? Because you probably are refreshed on that.
So
Yes.
carbohydrates and proteins. These are both organic molecules. I would say, no. Is that the term I'm looking for?
Yes.
Okay. So carbohydrates are used for short-term energy storage, and they're easily broken down. Now, there's three main types. There's monosaccharides, disaccharides, and polysaccharides. Now, simple sugars and things that taste sweet usually have monosaccharides, because they're easily broken down, and then disaccharides and polysaccharides are a little more difficult to break down, and therefore a little longer energy storage. Now, carbohydrates are made of carbon, hydrogen, and oxygen, and which are main components. And the difference between proteins and carbohydrates is that I believe proteins
Protein has
also,
nitrogen.
Yes, it's also made with nitrogen. Now, proteins, they're used for movement, structure, and nutrition, and it's a polymer of amino acids joined by peptide bonds.
Because proteins are made up of amino acids.
Yes. Alright, photosynthesis. Your thing.
Alright. So, photosynthesis. The original source is usually the sun. Sometimes chemicals like iron and Si. I don't know what Si is. Photosynthesis. Light energy is converted to chemical energy, and organisms that usually get energy from photosynthesis are called autotrophs.
Or organisms that produce their own food.
Yes. Thanks, Miguel.
And then
so,
there's that little diagram.
oh yeah, we have that diagram. So, water plus light it goes into the chlorophyll, and it traps the light energy to make food, and what it gives off is sugar and oxygen, and the oxygen is given off into the air, and the sugar is converted into starch, stored food in other parts of the plants. And then we have cellular respiration. Cellular respiration is the process of releasing the chemical energy in food to carry out bodily processes in the form of ATP. So what is ATP? Adenosine triphosphate. It's a temporary energy storage molecule. So a nucleotide adenine with two extra phosphate groups. The phosphate groups store compressed energy. It's charged with lots of repulsive force. So when moved, energy is released. So when the two phosphate groups are, I believe, broken, that creates energy.
Yes. So think about it like this. So there's adenosine triphosphate and adenosine diphosphate.
Yeah.
So ATP and ADP. When one of those phosphate groups, the reason why it's called triphosphate is because there's three phosphate groups. So when one of them is removed, it releases energy and is turned into ADP, or adenosine diphosphate. This is used for pretty much any sort of like cell movement. It's any biological, anything that's biological source of energy.
Mm-hmm.
Yep.
And hydrolysis of ATP releases energy. So water plus ATP results in adenosine diphosphate, one phosphate group, and energy. Energy is transferred to ADP or another molecule through phosphorylation. phosphorylation, gaining one phosphate group.
So from ATP to ADP to ATP to ADP. So back and forth.
Mm-hmm. So ATP can be used for chemical work, mechanical work, and transport work. And then through each work, it results in one ADP and one phosphate group.
Mm-hmm.
And then the 3 stages of photosynthesis. So energy is captured from sunlight by, I believe it's mesophyll, and inside the mesophyll is chloroplast. The energy is captured from sunlight. Light energy is converted to chemical energy in the form of ADP and NADPH. Energy in ATP and NADPH is used to make sugars from carbon dioxide. Can you explain NADPH?
NADPH is it's I can't remember. It's like I think it's kind of like a form of ATP. I don't think you really need to know what it stands for because I looked it up and it's like nicotinamide adenine dinucleotide phosphate, which is something we probably don't need to know. But
okay.
NADPH is kind of like, I think it's like secondary ATP if I'm correct. Probably
okay.
not.
And this formula, I believe the 2 reactants is water and carbon dioxide creates glucose and oxygen?
Yes.
If I'm not mistaken. Okay.
Yes, it is. Because it's 6 molecules of H2O, and then it's 6 molecules of carbon dioxide, which will make C6H12O6, which is glucose, and then it's 6O2, which is just 6...
and glucose is a carbohydrate, I believe, right?
Yes.
Glucose? Yes, it is. It is a simple sugar.
Alright, so these reactions happen in the chloroplast inside the mesophyll. So the light comes in and it uses water in photosystem 1, the electron transport chain, and photosystem 2, electron transport chain, and photosystem 1, and that creates ATP and NADPH and oxygen. And then the Calvin cycle uses carbon dioxide to create ADP. Can you explain this part, Jacob? The Calvin cycle or the Krebs cycle?
In all honesty, I did awful. I did awful on this. But
I haven't reviewed on the Calvin cycle yet.
I think photosynthesis is... Alright, correct me if I'm wrong. Photosynthesis is used to make glucose, and I think the Calvin cycle is used to break down glucose. No?
Honestly, I can't remember.
Let's search that up then.
Yeah, I'm on that.
Alright, Miguel can get back on that afterwards. So the chloroplasts are arranged into thylakoids, which are stacked into grana. This is where the light reactions occur. Stroma is the term for the area of the chloroplast inside the thylakoids where the dark reactions occur. So a dark reaction would be the Calvin cycle, which is without light. So in photosystem 2, light is absorbed by photosystem 2. Light absorbed by photosystem 2 is used to break up water molecules into energized electrons, hydrogen ions, and oxygen. And then the electron transport chain, high energy electrons from photosystem 2 move through the electron transport chain to photosystem 1. And then in photosystem 1, electrons released by photosystem 2 are energized again in photosystem 1. Enzymes in the membrane use the electrons to form NADPH. NADPH is used to make sugar in the Calvin cycle. Then hydrogen ion movement. The inside of the thylakoid membrane fills up with positively charged ions. This action makes the outside of the thylakoid membrane negatively charged and the outside positively charged. And then the ATP formation. As hydrogen ions pass through ATP synthase, their energy is used to convert ADP into ATP. Do you have a way to sum that up, guys?
Not really.
Let's see here.
I don't think I did too good on this section. I'm not
Yeah,
sure.
I've forgotten so much stuff. Okay. Well, if you have any questions, you can always ask Miss Henderson.
We'll probably end up looking at it later and
Yeah.
probably be able to explain it by then.
There is it tomorrow.
Yeah, there is tomorrow. We can always go back. So glycolysis. Glycolysis is, I believe, without oxygen, right?
No.
No,
Wait, no.
you're thinking of that's anaerobic.
That's anaerobic.
respiration, right?
Oh, okay, never mind. Glycolysis is the slow release breakdown of glucose into pyruvate or pyruvic acid. It occurs in the cytoplasm, and in the process, 2 ATP are produced.
So this is I was getting this mixed up with the Calvin cycle. Glycolysis is basically how we go from the glucose that's made from photosynthesis, and it's being turned into ATP through this process. Yes. So glycolysis happens, pretty sure, in all cells, and then photosynthesis happens in plant cells. Yes?
I can't.
Seems
confirm
logical.
that. Aerobic respiration. So aerobic is in the presence of oxygen. This creates much more energy. So 32 ATP is created versus 2 ATP. Than glycolysis or fermentation, which is anaerobic respiration. So in stage 1, the Krebs or the citric cycle, the pyruvic acid is broken down into carbon dioxide, and in stage 2, the electron transport chain, electrons from the Krebs cycle is used to make ATP. So the Krebs cycle, pyruvic acid enters the cycle, 3 carbon dioxide molecules are removed from the pyruvic acid, and 2 ATP and 2 electrons are released. Can you guys sum that up? Or
Jacob could do that.
Alright, so let's move on to electron transport.
We tried.
we tried.
That was good.
Electron transport. Electrons from glycolysis and Krebs cycle enter. The flow of electrons causes hydrogen to diffuse into mitochondria. The hydrogen build up inside the mitochondria and diffuse out through ATP synthase, making ATP. So the ATP synthase spins, and that creates ATP. Electrons cause hydrogen to join with oxygen, making water. Oxygen is the final electron acceptor. If O2 is not present, then the whole process stops. And then we have anaerobic respiration. Anaerobic is without oxygen, or the absence of oxygen, and there's 2 possible anaerobic pathways, alcoholic and lactic.
Isn't this why they is this why they suggest that you breathe when you're working out?
Yeah.
Because you use more energy. You use energy better.
You can use... Sorry?
Do we forget the water slide?
I know we have it on here.
It's like... Was it blocked, or...?
Sorry?
If you look at it.
On Google Docs, it's messed up completely.
Oh. Well, I have it here. I'll explain it. Alright, so these are the properties of water. Water is a polar molecule. One side is slightly positive, one side is slightly negative. It's efficient at transferring heat, and it's used in homeostasis. An example would be sweating. It is usually less dense when solid than when a liquid. It has a high cohesion, attraction between molecules of the same substance. It has surface tension, the result of cohesion. It creates a layer like a stretched skin over the fluid, so beads of water, or how a water strider is able to float on water. Adhesion is the attraction between molecules of other substances, so drinking through a straw, capillary action, and this is how plants get water. 7. They form solutions easily, components evenly distributed, so seawater and blood. Polarity allows it to dissolve both ionic and covalently bonded compounds, and it's a universal solvent. So a solute is a dissolvee, and a solvent is the dissolver, so the solute goes into the solvent.
I'll be honest, I took a biomed test a little while ago, and I got solute and solvent mixed up. So think of a lute, like the instrument, being able to go into a vent, but you can't put a vent into a lute. So lute goes into a vent.
Yes. Oh, that is a good way to remember it. Alright, so there's two types of reactions. Endothermic reactions and exothermic reactions. So endothermic reactions store energy in bonds, and the products have a higher potential energy than reactants. Example is photosynthesis. Exothermic reactions release energy in bonds, and products have lower potential energy than reactants.
And the graphs kind of show that too. Like an endothermic, by the end of the endothermic reaction, it has way more potential energy, and then exothermic, it's like releasing all of it, so its potential energy is lower.
Yes,
so if we think of with endo and exothermic reactions, remember that endo means inside, so all the energy is going to go inside of something. So it's going to absorb energy, and then exothermic is where it's going to release, so outside.
And then we have activation energy. It's just the energy required to start a reaction. And then enzymes. Those are protein catalysts that speed up reactions within an organism by lowering the activation energy. Jacob said before, they often end in A's. They're fast acting, 1,000 times a second. They're very specific and can break or make compounds.
Oh wait, and a good way to kind of visually represent that is the picture of the guy pushing the rock over the hill. With an enzyme, imagine that the hill gets like a bit lower, so it's easier for him to push it over. Yeah.
Oh, I actually have a picture.
Oh wait, it kind of shows that in the next graph.
Yeah. So you can see the activation energy required is higher without the enzyme, and with the enzyme it's lower. So how enzymes work. Enzymes act upon a substrate, the substance to be changed. On the active site, the area of the enzyme where the substrate attaches, the enzyme changes the substrate, breaking chemical bonds. And the enzyme plus the substrate are a very specific fit. Enzyme is affected by pH, temperature, and it changes shape of enzyme and is no longer a good fit. Alright, plants.
74 and 75 are blank, by the way. So
yeah, I know. I changed those on mine. It might have stayed.
Are we still doing plants, or is this the end of it?
We're doing plants today.
It's just the beginning.
The plants started it.
Yes.
We're actually doing plants today? Okay.
What's that picture?
Plants are multicellular. They're eukaryotes, autotrophs. They create their own energy. Cell walls of cellulose, and they can't move and have chloroplasts.
Obviously.
So there's 4 divisions of plants. Mosses have no vascular tissue. We'll get into vascular tissue in the next slide. Cuticle, which is a waxy layer that prevents water loss on the dermal tissue. And they don't have seeds either. Ferns have vascular tissue. They don't have a cuticle or a seed. Gymnosperms have vascular tissue, cuticle, and they have a seed, but the seed isn't a fruit. The seed is naked and is not in a fruit. And angiosperms have vascular tissue, a cuticle, and the seed is encased in a fruit.
Like in ferns, they have spores instead of seeds, right?
I believe on the underside of the leaf, yeah.
Yeah, those are spores instead of seeds.
Alright, and then there's 3 types of plant cells in ground tissue. We're going to cover the 3 types of tissue in a couple of slides. But how we remember it is metro PCS, and then it goes up a gradient of hardness. So parenchyma is outermost photosynthesis and gas exchange, storage, protection, and tissue repair. Collenchyma is middle. It provides support and flexibility. And sclerenchyma it's all over support, wood fibers, sclerids, transport, and vascular tissue.
Ravi?
Yeah.
Someone said, what about plants that move toward the sun? Because you said in the previous slide that they can't move, but technically they are...
By that
we mean like an animal is moving.
Like, plants won't grow some legs and just be like, oh, I think I'm going to go for a stroll today. It's a nice sunny day.
So they do sort of move.
They move, but not in the sense that they can just walk around.
Okay. They can't really freely move, I guess.
Exactly.
Yeah. Okay, so the 4 types of tissues in plants. There's dermal, which is the outermost layer. Vascular, which transports water and nutrients throughout the plant through xylem and phloem. And meristematic, which is regions of dividing cells, like plant stem cells that cause the plant to grow. And ground or fundamental, everything else. Okay, vascular. There's 2 types of vascular tissue. There's xylem and phloem. So xylem, it transports water from roots to leaves, and it's a one-way street, I guess. One-way street. And then these are made up of dead tracheid and vessel cells, and they act like straws. And gymnosperms only have tracheids. Transpiration, the evaporation of water from leaves. So xylem's main job is to transport water from the roots to the leaves. And then we have phloem, which is the other vascular tissue, and phloem transports sugars throughout the plant. It can flow in both directions. It's comprised of dead sieve and live companion cells working together. Sugars enter and exit sieve cells by active transport, and this requires energy. And the excess sugars can be stored as a fruit, seed, corm, bulb, taproot, and rhizome.
By the way,
Okay.
rhizomes are things like ginger.
I have a way of remembering xylem and phloem. Between them, xylem is like an insane asylum. There's one way in. You're probably not going out ever. And phloem is a flow, which is flowing. It's 2 ways.
That's good to remember that part, but the main part about them is that like xylem is water and phloem is like nutrients and stuff. I'm not sure how you would remember that specific part.
Well,
But
for
it's good
like
to remember
xylem,
that.
it's dead. It's made out of dead, right? Dead tissues and stuff. Phloem is made out of technically alive. By
It's
what's the
dead
thing
and
called?
alive.
What's the thing called?
Dead sieve and live companion cells.
A companion self. There you go.
Yep.
So it controls it like a puppet.
That's creepy.
Yeah,
Alright.
it got creepy when I.
Dermal tissue structures. So, on the outside, the cuticle is a waterproof, waxy layer that coats external parts of the leaf, and it slows evaporation. Stomata, pores in the epidermis, gas exchange controlled by guard cells. So, the stomata, the way it opens and closes is by guard cells, and I believe the guard cells work based on water. If water is
I think
present
water,
in it.
temperature, and gas.
Okay. And then we have how plants grow. So plants have these meristems that produce new cells by mitosis in localized areas of plants. So that's in specific parts of the plant. So apical meristems are at the tips of the roots and stems, and they increase in height and length, which is primary growth. Intercalary is in joints or axials between leaves and stems, and these result in side branches, grass or flowers. And then lateral, this causes increases in girth, which is the width, right? Yeah.
Yes.
And that's secondary growth.
So a plant will grow taller more than it will grow wider.
And lateral meristems. So we have vascular cambium and cork cambium. Vascular cambium is between the xylem and the phloem, and it creates new xylem and phloem every year. New xylem is wood. I believe that's how growth rings are created.
Yes.
And then cork cambium is located between the phloem and epidermis, and becomes dark.
Bark,
bark?
not dark.
Oh, sorry. And then there's 2 types of wood, which is xylem.
You
Your
mean
sapwood.
Sapwood and hardwood, not.
Oh yeah, wood is xylem. And there's 2 types, sapwood and heartwood. Sapwood is the outer light colored layers, and heartwood is the inner layers that get clogged and can no longer carry water. They're darker color and stronger than sapwood.
Isn't heartwood like the older parts of the wood too?
Yes.
Okay.
And we have the structure of the leaf. So the petiole is what attaches to the stem, and then the blade is kind of like the whole leaf itself, and then the veins are what kind of look like human veins on the leaf, and then you have the midrib, which is the line that goes through. So this is where photosynthesis occurs, and pores for gas exchange are on the underside. And that's it for today, it seems.
Really?
Do
Yeah.
we want to cut it early, or
Yeah, we'll probably just cut it early. You guys want to take any questions, I guess?
Send the link to the people for the form.
Oh yeah.
Oh,
the
yeah.
people who are actually still here.
And Ravi,
Yeah.
do you think later on you could send me the PowerPoint, because
Yeah.
with the classification, heredity, and evolution, the PowerPoint isn't showing up. But I mean, we could cut it early if you want to, because I need to get ready for the
Yeah, we need to be
Robbie,
prepared.
send the link in YouTube
Yeah.
for the people in chat.
So if you guys
Mhm.
go to this link, Alex, can you post it? If you go to this link, you just type in your name, and if you're in Miss Henderson's class, you'll get extra credit.
Wait, I can't post that in the...
Let me try.
Yeah, you try.
Comments can't contain links.
done.
Oh, it
Oh
can't.
wait, actually it says put it in the video description and
Oh.
refer to it. What? Okay, I got an idea.
You
Let
want
me
me
put
to
it.
do it?
No, I got it. I have it up
Oh.
here.
Okay.
I'm just gonna put it at the top just so it's easier for people to see.
Mhm.
Okay, if you refresh the page, you will see in the description a link, and that will lead you to the right page. And you just enter your name and press submit. Okay. So, do you think a PowerPoint's going to be on Learn, Ravi? Or I'm guessing that's probably where it's going to be at some point.
Yeah, we'll put it. We'll give it to Miss Henderson so she can put it up.
Okay.
Okay. Does anyone have any questions that we might be able to answer? No, we have to wait for the 45 seconds delay.
Yeah. By the way, guys, we've been having like it's a 45-second delay, so it's a little bit awkward, but we're trying to make it work.
Did they get the stuff for it?
It's in the description. You just got to refresh. I'll type it in case they are having issues.
You could also... Wait.
But tomorrow, I'm going to be explaining classification, so like binomial nomenclature,
Mhm.
heredity, evolution, organisms, populations, and ecosystems. So the stuff that we've been doing quite recently, like what we've been doing the past couple of months. So,
Mhm.
so any questions, anybody? Any questions? We'll take them.
Oh, and also, I think I said it before, but this video, I don't know how long it's going to take. It could take a while. It could take a very short time. Eventually, this will turn itself into a video, and you guys can watch
again.
it if you need to review anything or something. If I have the time, I'll try and kind of... I'm not sure if I can, but I think I can cut out parts that don't have information in them, you know, at the beginning and the end.
Mhm.
So